is share my screen with you and go over test 3. Now I think you can see it. It says test 3, closed book, starting with number 1. Everybody see that?
Yes.
Okay. Now, let me tell you about test 3. Those who got 80% or better, there were 8 out of 27 who took the test. So that's about a third. The high score for the test was 92. The average score was 63. So what this is telling me is that about a third of the class has a pretty good grasp of the material, but that means about 2/3 don't. And I understand that because this has been a really intense and dense set of subject matters, a lot of detail and a lot of integrating, making relationships from one system to another. So I'd like to do 2 things to try to rectify that. One is I want to go over the test carefully today, spend as much time as we need to make sure that everybody understands the questions, why they were asked, and what the answers are. And the second thing is what I've already announced, that I'm asking you to submit 2 questions by Saturday so that I'll have time to compile them and we can then spend as much time as we need on Monday to again go over this material and make sure you have a good grasp of it, everybody in the class. So let's start with the first question. In oxidation reduction reactions, which statements are true? Now, where did this question come from? I've indicated here a couple of the slides. We talked a lot about redox, but this question is really based on these 3 figures from lecture, showing how oxidation and reduction work in pairs. It's a coupled reaction, and how the electron transport chain, which you see in this figure, is nothing but a series of these things, of oxidation, reduction, handoffs. So which statements are true? Electrons are transferred back and forth between oxidized and reduced substances. Substance that gains an electron is reduced, but loses an electron, it's oxidized. And these reactions always come in pairs, like this. If one happens, the other one also happens. And finally, the function is release of energy for ATP synthesis. And when we do the next slide set, we'll spend a lot of time understanding how ATP synthesis happens. And a brief summary of it is right here, most of the ATP comes from the electron transport chain in respiration from the Krebs cycle. We're not going to talk about that now, but we will talk a lot more about it in the next slide set. Okay, is everybody good with this? Everybody understand the answers to these questions and why it was asked? Okay, no questions, then let's go to the next one about a graph. This is about reading a graph. Enzyme reaction rates are a function of temperature. That's illustrated in this graph. Answer the following questions. The y-axis is labeled rate of reaction. What does that mean? Well, it means that it's a speed function. It's the speed with which the substrate is converted to product. Then what is the reaction rate at 70 degrees? It's 0. Most of you got that, but not everyone did. With respect to the reaction rate, what's the significance of 0.2? That's the optimum temperature for this enzyme. In other words, temperatures below that are slower, temperatures above that are slower. And if you compare positions 1 and 3, which position reflects enzyme denaturation? And the answer is 3. If you get it above 50 degrees, the enzyme reaction rate slows down because at the higher temperature, it will start to denature. Okay, everybody good with that? Any questions about it? We've done quite a lot of graph reading in the course. And just one example of a graph is this one. How can the rate of an enzyme reaction be changed? By changing either the temperature, the pH, or the salt concentration. Number 3. What cellular processes can introduce variation into genomes of living organisms? Well, the ones here in red are the ones that are possible. Mutation, crossing over, random assortment, and fertilization. Okay, why is this important and where did it come from? It's best summarized in this particular slide. 3 sources of genetic variation without mutation. In other words, the given here is that a good source of variation is mutation. But beyond mutation, what else is there? Well, meiosis. Why do we have sex? Sex is one of the best sources of variation that can be introduced into a population. So sex is meiosis. And in meiosis, there are 2 sources of variation, crossing over and independent assortment or random assortment. So let's look first at crossing over. If you remember when we talked about this process, we mentioned tetrads. And there is a particular significance to tetrads because when they form, chromosomes are in very close contact with each other. And when they're in that close contact, they can actually exchange parts with each other at points called chiasma. And when they exchange parts, what you end up with at the bottom of this figure is both wild type chromosomes. You start out with 2 blue and 2 red. You end up with a blue and a red, and then 2 recombinant chromosomes that have exchanged parts. So you have a blue one with red tips and a red one with blue tips. So that is variation that results from crossing over. This only occurs in meiosis 1. There are 2 meiotic divisions. Only in the first one do we get tetrads and crossing over. In both of the meiotic divisions, we have independent assortment. That simply means that in metaphase, when the chromosomes line up in the center of the cell, the chromosomes homologous pairs have two members each, and the members can go, one member can go to either side, and whichever side that one goes to, the other one will go to the other side. And they do this on an entirely random basis. There's nothing influencing which way they go. So again, if you look at the diagram on the left, you can see starting out with red and blue chromosomes, one possibility is that you end up with nothing but red and blue chromosomes. But if they switch positions, as you see in possibility 2, then you end up with 4 different combinations relative to the starting positions. So this is the second way that we can get variation in meiosis. And it can happen at either of 2 or both, either or both of 2 different phases. Then finally, another source of variation is fertilization. When an egg is fertilized, the choice of sperm is entirely random. So out of millions of sperm available, only one actually fertilizes the egg. And remember that each egg is a gamete. Each egg is genetically unique. It's a result of recombination. And the same is true for each sperm. So when fertilization occurs, that's a source of variation. If you think about 2 people having kids, if they had 10 kids, each one of those kids is unique. And that's because for every one of them, there was a random fertilization event. Okay,
Professor.
Yeah.
This is Katrina Vibbard.
Uh-huh.
For both independent assortment and crossing over, can that occur to all 23 chromosomes, or is it specific chromosomes
No,
that happens to?
Yes, you're right. That can happen with any of the 23 pairs of somatic chromosomes.
Okay, thank you.
Okay, anything else? Now, the next question, only a couple of people actually got all of these correct. And I'm concerned about that because of this slide. All of these matching pairs came from this slide, which is a summary at the end of the Mendelian inheritance slide set. And I made a statement here that in order to think about genetics and discuss genetics, you have to know the language of genetics. If you don't know what the terms mean, you get bogged down quickly, losing meaning. So I've got definitions of all these different terms that are all important in discussing genetic inheritance.
Professor, this is Bennett.
Uh-huh.
Yeah, I got only partial on this. And I did refer to the slide like you had suggested, and I didn't see the back cross.
The back cross
Yeah.
is missing. Yes, you're right. The back cross is missing. Okay, does anybody know what a back cross is?
Hi, Professor, this is Aiden. A back cross is when you breed offspring with a parent.
Right. It's exactly what it sounds like. It's a back cross. So if you take an F1 progeny, cross it back to the parent, that's a back cross. In contrast to an F2, where you take an F1 progeny and cross it to another F1 progeny. So the distinct difference between a back cross and an F2. Thank you for pointing out that that term is missing from this list. But for those of you who haven't solidified in your minds these definitions, please do, because it's important and it will very likely come up again in subsequent quizzes or tests. Now the next one is another one about graphs. Enzyme activity is a function of both pH and temperature. From the graph on the left, describe what happens to the enzyme's activity as its environment varies from pH 5 to 10. Some of you got this right, but you didn't really, many of you didn't really describe what's happening. The enzyme activity is increasing between 5 and 8. 8 is the optimum, and then it decreases from 8 to 11. To get full credit on that, you had to say that there's both an increase and a decrease shown by this curve. And then the second part, from the graph on the right, indicate the color of the arrow pointing to the temperature at which the tertiary structure of the enzyme is intact. And that is the red one. Above 40 degrees, the temperature is too hot for the enzyme, the rate slows down and the enzyme starts to denature. So the tertiary structure is intact at all temperatures up to 40 degrees. All right, we had several breeding questions. And we spent a lot of time going over examples of how you do these. And some of you clearly know how, but there's more of you who don't. And it's important that you do know how to solve these problems. And they'll come up again. If not on the test 4, then certainly on the final, there will be more questions about inheritance. So if you're not clear about how to do these, make sure you understand it. And the first thing you have to do in trying to understand is to read the question carefully. Look at all the words. So this one says, a true breeding tall pea plant is crossed with a true breeding short pea plant. What does true breeding mean?
Justin Hansen. The true breeding means like both are the same, have the same genomes or same genes.
Yeah.
So like double, yeah.
Homologous?
Homologous,
Yeah.
Good term to describe a true breeding plant. So a true breeding plant means that it's been inbred, crossed with itself for many generations so that all the loci are homologous. So in a diploid, there are 2 copies of a gene at every locus. And after inbreeding, both of those copies of a gene are the same. And so a true breeding tall plant means that it's homozygous at the tall locus. So the other thing you have to do with a problem like this, there's no symbol given for the locus you're talking about. So I've arbitrarily assigned a letter P as a symbol for tall. And the question says tall is dominant to short, which means that that short is recessive. So since it's true breeding and dominant and tall is dominant, then the dominant genotype is big P, homozygous dominant. That automatically makes the alternate allele, little p, little p. The parental cross then is big P times little p. So the F1s have only one genotype, only one possible genotype, and that's a heterozygote. So they're all heterozygous, and since tall is dominant, the phenotype is all tall. But what gametes can the F1 produce? Well, either big P or little p. So to go to the F2 generation, which is what the question calls for, the clearest way to do that is to plot out a Punnett square. Both parents. On one axis, you have a parent big P has gametes big P and little p. The other parent is the same. It has gametes big P and little p. So if you put it together, the combinations, you have one big P, two heterozygotes, and one homozygous recessive. So to answer the question, the genotypes are 1 to 2 to 1, one homozygous dominant, two heterozygotes, one homozygous recessive, and phenotypes would be 3 to 1, 3 tall to 1 short. So this is about the simplest type of cross that you can conjure up. It's called a monohybrid cross because there's only one locus involved.
Professor,
Yes.
when for the future reference, when we're putting these answers, do you want us to put like the ratio like that, like gametes to gametes and phenotypes to phenotypes, or just what comes out?
If they're called for.
Okay.
So for example, one of the questions coming up, all it asks for is gametes.
Oh.
So if it's asked for gametes, that's what you put. In this case, it's asking for genotypes and phenotypes of both F1 and F2. So that's what you should put down. That's why I say read the question carefully, make sure you understand the information it gives, and then what is it asking for? Anything else about how to do a monohybrid cross? All right, next question. Choose correct statements from among the following. All enzymes are proteins. Not all proteins are enzymes, that's true. Certain enzymes are active only if they have a partner. And this refers back to this slide. And we talked about enzymes. This is dedicated just to enzyme partners. There are cofactors, coenzymes, prosthetic groups. Most enzymes don't act alone. They have to have a partner. And finally, enzymes with quaternary structure are composed of more than one polypeptide chain. That is true. Okay. Any question about number 7?
I had a question on that.
Yeah,
I got all 3 of those right, but it said I got 2 out of 3.
all right. That's a good question. I will check on that. Kaylin Hilberg. That's question 7. All right, I'll check on that and let you know what I find out.
Thank you.
All right, the next question is about the order of events in mitosis. And that refers specifically to this slide, steps in mitosis. describes each step as it's been defined. And so what I did was write down all these steps and then scramble them, letting you then pick out the one that isn't scrambled. And that turns out to be the first one. Chromosomes condense, line up at the center of the cell, get pulled to opposite poles, arrive at the poles, and then the cell divides. And the other three are all perversions of that. And the cell divides at the end. We have a question coming up on cytokinesis. I think it's the last question in the set. The difference between cytokinesis and animal and plant cells. In animal cells, there's a contractile ring that squeezes the cytoplasm until two cells emerge from one. This can't happen in plant cells because they have a wall. So what happens is that a new cell plate, a new wall forms between the daughter cells, fuses with the wall, and it's only then that the cell can split into two daughter cells. So this is the answer to that final question that we'll come to later. And now we have another genetics problem. Color blindness. It's sex linked. A male who is colorblind has children with a woman who is a carrier for color blindness. So you read that and you say, okay, if this male is colorblind and it's sex linked, then the mutation could be either on his X or his Y. But then you see the next sentence says, the woman is a carrier for color blindness. That means that the mutation is on an X because females don't have a Y. So now we know the male genotype. I use a star for the mutation. The male is X star Y. And he's colorblind because he has this mutation. Now, what gametes can he produce? He will produce half X stars and half Ys. Now, the female we know is a carrier. And what is a carrier?
Someone with the allele?
Has an allele, yeah. What else about a carrier?
They only have one because it's a recessive gene. It's not dominant.
In the case of sex-linked, dominance isn't really a factor. So a carrier, what does it mean?
Maybe like instead of allele, like a chromosome, like that person has that genetic chromosome, so the likelihood could be passed down.
Well, if they have a mutation, it can definitely be passed down. The term carrier is designated for somebody that has a mutation, but they don't have the disease. So they're carrying the mutation, but they're not expressing symptoms. So because this woman is described as a carrier, we know immediately that her genotype is X star. She's got a mutation, she's showing no symptoms, so her second X doesn't have, I should say the first X, one of her X's doesn't have a mutation, the other X does. So what gametes can she produce? Either X or X star. So if you plot these out, the male X star Y and the female X star, what do you get? X star and X star, these are both females. They're both XX. One of them is a carrier because she's only got one mutation. The other one is colorblind because both of her X's are mutated. Now the other two progeny, possible progeny, are boys. One is XY, he's not colorblind. The other one is X star Y, he is colorblind because he's only got one X and it's mutant. So half the progeny are boys, half are girls. Half the boys are colorblind and half the girls are colorblind. So the answer to the question is 50%.
Professor,
Yeah,
would color blindness be the phenotype?
yeah.
Because I thought phenotype was more just like a physical characteristic.
Yeah, it is a physical characteristic. You're saying, how can you see colorblind?
Yeah.
You look at somebody, how do you know they're colorblind? You don't. So this, I guess you could regard it as a special case
Okay.
phenotype. Contrast that with the genotype, which is a symbol. A phenotype you describe in words. So
Okay.
Colorblind is the word description. X star is the genetic description.
Thank you.
Okay, anything else about color blindness or sex linkage? Sex-linked traits are genetically different from autosomal mutations. The next one, number 10, match each phase of mitosis with its correct description. And this goes back directly to this slide, where you have on the left side a description in words of each phase of mitosis. And what this question is asking you to match up the word description with the phase. So you can you can do the matching up yourselves. Telophase, you arrive at the poles. Interphase, DNA replicates. Cytokinesis divides. Prophase, you get condensed chromosomes. And usually when there's a matching exercise, it involves the best choice of answer. That doesn't mean that that any given answer is the only one. It's looking for the best one. Now the next question, this is something that's dear to my heart, as some of you have recognized. Mendel was a genius in that he had two really seminal insights. He did a lot of things, and many of you answered this question by telling me some of the things that Mendel did, and that's all great. But those things were not what made him unique. What made him unique was that before he started doing crosses, he developed inbreds, and once he started doing crosses, instead of stopping with the first progeny that he got, he crossed these progeny to each other to get the F2 generation. And that's where he first saw a recessive trait. And that's where we get the concept of dominance and recessive. Dominance is what he sees in the F1 generation. Recessiveness is what he sees in the F2. And because I think this is so important, I emphasized it over and over. And in fact, on this summary slide, right at the very top, keys to Mendel's success: inbreeding and the F2 generation. Some of you used those 4 words, and that was perfect. Okay. Chemiosmosis. It's a critical step in ATP production. We're going to talk a lot more about that next time when we deal with respiration. Indicate which items are true about chemiosmosis. It generates ATP by passing electrons across a semipermeable membrane. And you see that in this illustration. Here's the membrane, here are the electrons going across it. This last step is the chemiosmosis part. The source of electrons is the electron transport chain. That's shown here on the left. You have these proteins embedded in the membrane that pass electrons one to the other until they finally get to the last step, which is chemiosmosis. It's anabolic. Why is it anabolic? What is it? What does chemiosmosis do? Any thought? Well, it makes ATP from ADP and phosphate. So that's building up, and it takes energy. It takes energy from electrons and makes chemical energy in the form of a high energy bond in ATP. So it's an anabolic process. It requires an electrochemical gradient. You can see that shown well in this slide, where the concentration of electrons is much higher on one side of the membrane than the other. And when they pass through the membrane from higher to lower concentration, ATP is generated. And it can be found in both mitochondria and chloroplasts. This has been mentioned already in the course, but we're going to talk a lot more about its occurrence in mitochondria when we talk about respiration. So anybody have a question about electron transport chain and chemiosmosis? Chemiosmosis is the last step in electron transport. Now, what event leads to a diploid cell in a life cycle? Well, if you refer to this figure of human reproduction, right at the top is an egg and a sperm. Now we know by definition that they are the products of meiosis, and therefore they are both haploid. If the sperm fertilizes the egg, that's called fertilization step, and now you have a diploid zygote. And from that point on, humans remain diploid. The only haploid part of their life cycle is in the testes and the ovary, where gametes are made and meiosis occurs. So the answer to the question is fertilization. The next question is sort of indirect, but what I was really trying to get at is your understanding of the electromagnetic spectrum and what it means. So the question is, you're out and about on the road to Ramona and you notice plants that aren't green. So the question is, how do you explain plants that aren't green? In this case, the plant is orange. One possibility is that it's a parasitic plant. Another is that it's really a green plant with an orange pigment. Another possibility that it's nutrient deficient. And this goes back to the electromagnetic spectrum, which you recall in the spectrum, there's just this one very small window of wavelengths that we can see and that plants can perceive. And within this little window, the wavelengths vary in color from red to blue. Now, we can see both of those and everything in between. Plants can see only, and by see, I mean absorb the energy of only red, yellow, and blue wavelengths. They cannot absorb green, and so they reflect it. So when we look out at plants, usually what we see is green, but not always. So you might see, for example, a red tree, an ornamental red maple. Now, is that a red tree or does it have chlorophyll? And the answer is it has chlorophyll. It's perfectly good at photosynthesis. It's just that it makes another pigment besides chlorophyll, an anthocyanin, and that pigment happens to be red. So it masks the green. So that's a possible explanation for the orange plant, that the orange plant is really a green plant, but in addition to chlorophyll, it makes an orange pigment. It could be that for this particular plant, the soil is very poor, and so it's nutrient deficient and it turned yellow. Some people said it might be an albino. I didn't accept that for the following reason. First of all, an albino plant is pure white. And secondly, after the resources of the seed are exhausted, the only thing that can keep an albino plant alive is if you feed it sugar. Well, out in the chaparral, it's very unlikely anyone is coming out every day to feed this plant sugar. So that's not a reasonable explanation for seeing an orange plant. If it were in the fall and leaves were falling, it could be a deciduous tree whose chlorophyll has been degraded because it's that time of year. Or finally, and these are actual pictures from the desert east of San Diego, you see a green plant here with a yellow patch. This is a parasitic plant called dodder up close. And the way it gets its nutrition, it has no chlorophyll at all. It gets its nutrition from sucking juices from a green plant that can do photosynthesis. So that is the final possibility to explain why you're seeing an orange plant. It's possibly a parasitic plant. It could be a green plant with an orange pigment, or it could be an orange plant that's nutrient deficient. Now, a lot of you didn't even try to answer this question. Not sure why. But whenever you're faced with that, you feel like you're blown away, stop and take a deep breath and think about what you've learned in the course and how it's relevant, how it might be relevant to the question that's being asked. So instead of leaving something blank, put down everything you can recall that's related to that. Okay, enzymes in a cell are mediating thousands of chemical reactions at any time, but they're very fragile. They can be denatured. What are the things, changes in the environment that might denature them? We've talked about these before: pH, temperature, and salt. There are numbers of slides in the set that relate to this question. I chose just one to illustrate it, where it says in words, temperature, pH, and salt concentration. And it shows, it illustrates here a a normal protein with its tertiary structure intact. And if it's abused by high pH or temperature or ionic strength, the tertiary structure will break down to just a linear polypeptide, which has no enzymatic activity at all. The next question, match each of the following terms with the most correct option. Dominance and complete dominance and complete dominance. There's a slide in the deck that says that explicitly. So heterozygotes display both phenotypes, that's codominance. Incomplete dominance is when heterozygotes display a blended phenotype. You remember the cross between a red chrysanthemum and a white one. The F1 was pink. That was an example of incomplete dominance. Complete dominance is when you only see the dominant phenotype in a heterozygote. And enzymes increase the rate of reactions by lowering activation energy. And what I wanted you to think about in this question was exactly how do enzymes do that? What is the mechanism by which they lower the activation energy? So I chose, we had a number of slides about this, but I chose this one to illustrate the point here. And this is where this question came from. So what statements in this list indicate actions that an enzyme or that occur as an enzyme is converting substrate to product? Well, one is the binding of the substrate to the enzyme active site. Another is the enzyme changes shape. And when it does that, the bonds in the substrate are broken or weakened. And after that happens, the products are released. And that's illustrated here, where you see the enzyme in orange, the substrate is green. Their shapes are compatible, so they fit together. And once the substrate is in the active site, the enzyme changes shape. And you don't see that here, but you do below. Here's the binding of the substrate in the active site. Here the enzyme changes shape, and in so doing, it changes the shape of the substrate, weakening or breaking some of its bonds, so that the last step is when the enzyme releases the substrate, releases the substrate to products, it's now broken into two. It started out as one object and it ends up as two. So those are the steps that I was looking for when I asked this question. And I wanted you to think about this because it's one thing to just say superficially, oh, an enzyme is an organic catalyst. It converts substrate to product. But how does it do that? Well, these are the steps that an enzyme takes. And this next question is about regulation, and I wanted to emphasize this because we are full of enzymes. We have thousands of enzymes in our bodies at any point in time. Most of them are silent at any point in time. Only a few enzymes in our bodies are actually working. So what determines whether they're on or off? I listed some of the possibilities here on the left, And I wanted you to match up with how that mechanism works with something on the right. So feedback inhibition, the end product blocks the pathway. Reversible inhibition, competitive and non-competitive, and irreversible inhibition. And those come back to these illustrations. Reversible inhibitors can be competitive or non-competitive. A competitive inhibitor mimics a substrate. So it has almost the same shape as a substrate, but not quite. But it's close enough that it can fit into the active site of the enzyme. And while it occupies that site, substrate can't get in. So there is no enzymatic reaction because the substrate can't get into the active site. So the higher the concentration of inhibitor, the fewer enzymatic reactions there will be. A non-competitive inhibitor doesn't compete with the substrate for the active site. It has a different site. And when it binds to that site, it changes the shape of the active site. So now the substrate doesn't fit at all. So again, if there's a high concentration of this inhibitor, then most of the active sites are going to be closed and be very little enzymatic reaction. And then there's feedback inhibition, which is a very effective way of achieving homeostasis in a system. This, as you may recall, many of you didn't recall this, there's a pathway, starts with a substrate, goes through a number of steps, ends up with a product. And I made the analogy here with a wound that you might have. Say you cut yourself. And you need to immediately start healing that wound, and you need a particular protein to form a scab. Well, as soon as you cut yourself, this pathway starts running at full speed, producing a lot of this protein that you need to make a scab. But then you heal, and as you do, the need for this scab protein becomes less and less until finally you're completely healed and you don't need any of it. And yet the pathway is still running, still making this end product. So what's happening is that the end product accumulates, but there's no use for it. But in this type of a system, the first step catalyzed by enzyme 1 has a site to which this end product can bind. And when it does, it inactivates enzyme 1. So now if the first step in the pathway can't happen, none of the others can happen either. So binding, this end product binding to the first step shuts down the pathway. No more end product is made because you don't need any more to heal your wound. And so this pathway then would remain completely shut down until the next time you need a scab. So is that clear how feedback inhibition works? It's a very sophisticated way to fine-tune enzymatic activity. Okay, no questions about that. And the reason I'm going over these slowly and carefully is that, as I said at the outset, about 2/3 of you missed a lot of these questions. That means you didn't know the answer. And so I'm trying to do two things here. One is to tell you what the answers are, but secondly, to prepare you for the next time. So you'll know how to look at a slide set and familiarize yourself with the information so that when you face a question on the test, you'll have a resource to get an answer. Okay, the next question, number 19, It's about my former student and his work on mitosis. He was interested in mitosis, but before starting out to do an investigation, he read the literature. And he made many observations, including the fact that chromosomes move long distances in dividing cells, relatively long, and that there are certain proteins called motor proteins that are associated with subcellular movements. And a particular one of these, called dynein, is present in cells when they divide. So he wanted to get a conclusive answer to a question about the role of dynein in mitosis. And so what I asked was, what was his experimental approach to ask conclusively, what role does dynein have in mitosis? So he inactivated the dynein encoding gene, and then he compared the mutant with wild type. So just to remind you, this is mitosis, this is metaphase and mitosis, and these are the chromosomes all lined up in the middle of the cell. And these little walking figures represent dynein proteins, motor proteins. And each one is walking toward the pole of a cell to the right, or walking toward the pole of a cell on the left. So what Satoshi asked is, what if I took this away? What if I made a cell that didn't have any little motors? What would happen? So the way he approached that was, this is pre-CRISPR technology, but still targeting. So he knew the sequence of the dynein gene. He made a vector that would specifically disrupt that gene. I won't go into details of how that was done, but you end up with a mutated dynein gene, which is totally dead. Has no function at all, no motors. And he did two things. He first just looked macroscopically. He was working with a fungus, so this is a microorganism. He looked at his petri dishes, and then secondly, he looked microscopically. When he looked at petri dishes, he could see that wild type grew fast, and here's his mutant, this poor little sick mutant that can just barely grow. It takes weeks to make a little colony that big. You may recall I told you that for months, Satoshi was throwing away his mutants because they didn't grow enough that he could even find them. You had to wait for weeks at a time and cut away the control or the non-mutants so they didn't overgrow his mutants. And then you can see in the plate on the right, the little dynein mutant can still grow even in the presence of drug, and that's because it has this drug resistance gene. Then when he looked at a microscope, I won't go over all the things he saw. The most important thing I want to point out here is that when he looked at the microscope and he asked in living cells, what's happening to the nuclei? Well, in wild type, nuclei move really fast. In a mutant, they hardly move at all. So that is why there's such a little bit of growth here, is that mitosis is just barely happening. There are very few cell divisions because there are no motors or very few motors. So that allowed Satoshi to define conclusively what's the role of dynein in mitosis. Then I asked, what's the relationship between Darwin and Mendel? On the very first slide of this set, I had indicated it. that Darwin established natural selection as a force controlling speciation, and Mendel provided the rules. The rules for variation that underlie natural selection. So what I was looking for in an answer here is anything to the effect that meiosis generates variants and natural selection sorts among them to find the most fit. At what stage in meiosis does the nuclear condition change from diploid to haploid? Well, that was a straightforward question, and the answer is in this slide. Meiosis 1, the first 3 phases of it are all diploid, and then at telophase 1, it becomes haploid and remains haploid then throughout meiosis 2. So the answer to this question is, where does it change? It's in telophase 1. And then we had a question about work and entropy. A, work is a term used to describe matter moving in a particular direction that it would not have moved if it had been left alone. And entropy is a term used to describe matter moving in a random direction. And those answers or those questions were illustrated in these slides. What is work? And what is the second law of thermodynamics? Any question about that? Pretty straightforward. Okay, another inheritance problem. What you're given here is very simple. A parent that's homozygous at 2 loci and one that is heterozygous at both loci. So we have big F, little g crossed with big F, big G, little g. Now this question is only asking for gametes. What gametes are produced by the homozygous parent? Well, it only has big F's and little g's. So the only gametes it can produce are big F, little g. What gametes are produced by the heterozygous parent? Well, you have 4 possibilities: homozygous dominant, heterozygotes, and homozygous recessive. So those are answers to the first 2 questions. Then how many boxes would you need for your Punnett square to account for all possible gamete combinations? Some of you didn't know. Some of you said 16, and some of you said 4. The correct answer is 4, and the reason is this. Since the homozygous parent can only produce one type of gamete, you don't need 4 boxes across the top because those 4 boxes are all the same. If you filled in the Punnett squares, everything would be the same from the first column on. On the other parent, you do have 4 boxes, but if you do all the possible combinations, you only need 4 boxes. So 16 boxes is not incorrect, but if you said 16 boxes, it didn't indicate that you really understood what the question was asking.
Can you elaborate on that further, Professor? Like why you wouldn't put more boxes for the homozygous parent?
The short answer is because they're not necessary.
Okay.
So just imagine, I can't really show you, but here in this box, you've got the only gamete that parent can produce.
Oh, because you get the same results if you had 16 versus 4.
Right, so you can make another column here, would look exactly the same, another column exactly the same, and another column exactly the same. So you could make a 4x4, but you don't need it. And what I was looking for is that if you would actually recognize that you don't need 16 boxes, you only need 4. to account for all the possible gamete combinations, which is what the question is asking for.
Okay.
Okay, yeah, it's good to think about these questions and ponder them, because Mendelian inheritance is a good mental exercise. Okay, the next question actually came from a worksheet, which reminds me to tell you that when you study, you should review the worksheets as well. This one was from that worksheet. It had the overlapping, the Venn diagram, the overlapping circles, and it asked for what processes are unique to meiosis, which are unique to mitosis, and which are common to both. So this question is, indicate items common to both meiosis and mitosis. And I've circled them here from the test, and here they are from the worksheet. And finally, we've already answered this question about cytokinesis in animals and plants. I showed you a picture way back where, the difference here. Animals, you have a contractile ring, and I was looking for that term. In plants, you have a cell plate, which fuses the cell walls of the two daughter cells. That's the end of the review of test 3. And as I said at the outset, about 2/3 of the class struggled with a lot of these questions. And that's why I wanted to go over the test in detail with you. And also, I've asked you to submit questions for us to discuss on Monday. Well, while all this is fresh in your mind, I'd like to have a full period class discussion of whatever you're having trouble with or whatever you'd like more information about. And I've used all the time for this class period. So I'm going to stop sharing. And anything else before we go? Next class period will be on Monday. And in the meantime, make sure you look in your announcements. There's a new announcement that has a lot of information in it about upcoming schedule.
Thank you for the 2 submissions. I feel like I would have really struggled with the time parameters of the test if there were not the 2 submissions.
Oh, the 2 attempts?
Yeah, I felt like I needed that entire, what was that, 2.5 hours to do
Yeah.
that.
Yeah, I had initially, my thinking was about that you don't really need 2 attempts, but the more I thought about it, the more I thought that 2 attempts could be part of the learning experience. And you try it and you have a little time to think about what you didn't know, and that reiteration in your mind helps you to learn the material.
Exactly.
So I've changed my mind about double attempts.
Thank you.
Okay, anything else before we go?